with because they're really so small. Also, their biology is virtually unknown. Although a few species have been shown to be predators of other minute insects, we really don't know very much about them. And they're certainly of no economic importance. So all this should have probably actually discouraged me from wanting to study the systematics of dipsos, as we call them, but it really didn't. And this is mostly because even before we started to work on this group, for real, we suspect that there may be substantial undescribed diversity in a group, phylogenetic relationships between and within the families of dipsos were unknown. Minute littlebugs were thought of as being part of the early diverging lineages within the true bugs, although their exact phylogenetic position was not clear. And finally, and this is really the reason I wanted to study them, some of them are really quite cute, like this Nanochorus on the lower left. Others have simply outrageous morphology, such as the species of Amatitis on the right, where the male has a strongly modified labium, which is part of the mouth parts with a little grasping structure on the outside, which is something that's completely unheard of in true bugs. So we were lucky to attract some funding in 2013, and we've been studying the group ever since. I want to start with a few slides on the biodiversity of dipsos. From what we can tell after working on the group for several years, dipsos are probably not as megadiverse as some groups of tiny flies or wasps, but it is clear that there was and still is substantial undescribed species level diversity out there. We started with about 320 described species in 2013, and we have slowly worked our way up to around 552 at the moment. As I just mentioned, dipsos are tiny and are often associated with leaf litter, the canopy of trees, or other difficult to search microhabitats, including gravel banks along streams. The most effective methods to collect them are passive trapping methods, including leaf litter sifting, canopy fogging, or light traps up in the canopy. Because there are so few of them in the curated parts of collections, they've always been thought of to also be really quite rare in nature. And their curation is really quite awkward, and they may have to be slide mounted, which is something true bug people don't really like to do. But you can also see a typical drawer here in the middle photograph, where there's one single little specimen representing one species in a big unit tray, because it's really the only known representative of its species worldwide. So what we did starting in 2013 is take advantage of residues from passive trapping projects deposited in many museums around the world. We sorted through these jars of insect soup, as we call them, and in relatively short time, we pulled more than 24,000 specimens of dipsos out of these samples, many times the number of curated specimens that were at that point held in collections. The chart below traces the number of species of dipsos described over time. And as you can see on the right, the taxonomic activity from our lab and with collaborators has started to make an impact on the number of described species. We have mostly focused on documenting and describing species in the largest and morphologically most diverse family, the Schizopteridae, from the New World tropics. And for each genus or subgenus we have revised, we have multiplied the number of described species, as for example, for Guapinanus on the lower right, where the count has gone from 1 to 20 described species, or the Schizoptera subgenus Cybirflaps, where numbers are in the process of going from 3 to 39. There's lots of work left to be done, as you can see from these 4 genera genus groups, where we have fairly accurate estimates of yet to described species based on our current samples. And there is lots more undescribed diversity in the Afrotropical and Oriental regions. While our approach to describing this diversity has often incorporated molecular studies, we have mostly relied on morphological features to diagnose species. And this makes sense for most schizopteridae, where males are frequently collected. Male genitalic features provide excellent species-level diagnostic features and can be documented fairly efficiently using a combination of microscopical techniques and even really just straightforward, simple line drawings. There are some exceptions to that. For example, the genus Teniophyes on the right, where males are rare and diagnostic features of the females are somewhat limited. And this is where we're relying to a greater extent, although not exclusively, on molecular data to delimit species. This was really quite different in the second species-rich family, the Ceratocombidae, that are your classic boring little brown bugs, because from what we can see at the moment, genitalic features are relatively little used to delimit species. They're found worldwide, including in temperate regions. There's 40 described species out there. And about half of the samples we pulled from residue, meaning about 12,000 species, specimens, belong to this family, and they originate from about 70 different countries. Well, I'm pretty certain that we could probably dig up some morphological diagnostic features for many of these undescribed species. They are just not very exciting to look at. But I think that one way of making progress with this particular group would be to mass barcode sequence the samples we have and then use spinning approaches to help delimit species and then do more targeted morphological exploration. Moving to phylogenetics, I mentioned that we knew next to nothing about phylogenetic relationships between the families of minute little bugs and even less about relationships within the largest and most diverse family, the Schizopteridae. Several phylogenetic hypotheses across all 7 true bug infraorders have been published during the last 4 years that are based on different data sets ranging from Sanger and morphology, as the one I'm showing here on the left, to molecular-only data sets that are derived from ultra-conserved elements or transcriptomes. And they all show that dipsos are part of a clade that includes the water strider, the geromorpha and relatives, and the unique headed bugs, the Onychocephalomorpha. And dipsos are recovered as sister to Onychocephalomorpha in most of these studies. Dipsos have traditionally been subdivided into 5 families, the Ceratocombidae with 2 subfamilies, Schizopteridae with 3 subfamilies, and the smaller families. The numbers are described species as they were in 2013, the Dipsocoridae, Hypsipterygidae, and the monotypic Stemocryptidae that are really only known from the type series. We assembled a decent-sized concatenated molecular and morphological data set comprised of 133 in-group taxa with species of Geromorpha, Onychocephalomorpha, and Nepomorpha, the aquatic bugs, as outgroups. All recognized families, subfamily, and tribal-level taxa of dipsos were included, and we had dense genus-level sampling of the morphologically most diverse family, the Schizopteridae, including recently described and still undescribed genera. We struggled a bit with the fact that some of the dipsos were derived from relatively poorly preserved residue samples, but by using a combination of Sanger and next-gen sequencing methods, we were able to generate a molecular data set with pretty good taxon representation. We coded 159 morphological characters and included them in combined analyses. This also allowed us to incorporate a species of Stemocryptidae, for which only the types are known. They could not be sequenced because they're slide mounted. Our morphological character exploration built on the excellent comparative morphological studies that had been done by Wygosinski and Stiss in the mid-2000s, in the mid-20th century. But we also did substantial exploration and documentation of morphology on our own, in particular on male, to a certain degree, female genitalic structures, but then also really wing venation, where there were lots of puzzles to solve in terms of wing vein homology. The final matrix was comprised of about 30% characters from the genitalic structures, followed in numbers by characters derived from the forewing and the head. While we explored molecular and morphological data sets in separate analyses, what I'm showing here are the results of the combined morphological and molecular analyses. We ran both likelihood and parsimony analysis with both equal and implied weights. The tree on the left is the strict consensus of the best trees derived from 7 implied weights analyses with different k values. Overall, the topologies from all parsimony and the likelihood analyses were very similar, but one important thing is the placement of Stemocryptidae, the taxon that was only represented by morphology, differed between the different analyses. Dipsos, Schizopteridae, and Dipsocaridae are monophyletic in all analyses. Species of the Ceratocombat subfamily Trichotilloninae never form a clade with the remaining Ceratocombidae, and while 2 of the Schizopterid subfamilies are monophyletic, the third, the Argyriinae, are not. Based on these results, it was fairly straightforward to revise the high-level classification of the group. We are now recognizing 6 families with Trichotilloninidae raised to family status. We synonymized Argyriinae and Schizopteridae, and we are recognizing 2 subfamilies in the 2 large families, Ceratocombidae and Schizopteridae. Diagnoses for the families and subfamilies are derived from the parsimony and likelihood optimizations of the morphological characters. And while some of the diagnostic features may be a little difficult to see, the great majority, we think, is pretty accessible even to a non-specialist, as for example, distinctive adhesive pads that are found on a metacoxy that are one of the diagnostic features for the entire Enfer order, or striking dextral asymmetry of the male abdomen that is a diagnostic feature for the new status subfamily Trichotilloninidae. We have also used phylogenetic hypotheses to inform taxonomic decisions in many of the subfamilies and genus groups that we are revising. I have to admit that we have largely relied on molecular-only phylogenetic hypotheses for these projects. There's really no good excuse for that, other than that we're a little time constrained and we really wanted to move forward with documenting and describing the diversity in these groups. So, for example, in the Hypsilosoma this molecular phylogenetic hypothesis on the left provided some of the arguments for synonymizing Williams-Ockers and Amatitis. And then, of course, we backed that up with morphological data, and we then described a number of new species in that genus. As a second example, when we noticed the isolated phylogenetic position of the 2 species with orange arrows in the molecular phylogeny, we examined them a little bit more closely morphologically, and we found that it could be easily diagnosed as 2 new genera, and that's what we did. For another project, we used a combination of Sanger and NextGen molecular data to infer a phylogeny of the Carxidia genus group within Schizopteridae. One of the more exciting aspects of the study, I think, was that a lot of the molecular data were generated from point-mounted and dry specimens, including some that were really badly overgrown with fungus, and one that was a paratype that was collected in 1922. The main goal was to assess relationships among the currently described 5 genera and 18 species to inform taxonomic descriptions of the more than 150 undescribed species that belong into this genus group. We found that while 3 of the large genera are monophyletic, the largest, Carxidia, is not. And we proposed that 5 genera will need to be described to best accommodate this undescribed species diversity in the group. While we again did not perform combined analyses, ancestral character state reconstructions of selected morphological characters show that there are a few diagnostic features that are unique to a single clade, but all described and undescribed genera can be straightforwardly diagnosed by a combination of really a relatively small number of morphological features. Finally, what have we learned about the morphology of dipsos? Quite a bit, I want to say, but of course, there's lots more to discover. I here just want to highlight 3 areas that we found quite exciting. The wing type diversity across dipsos, head shape patterns in one of the genera, the genus Nanocorus, and enigmatic male-specific organs across Dipsacromorpha and specifically within the Choricidae genus group. And in most cases, it really comes down to the question if the different structures and modifications we see in these insects are homologous or not. Many species of true bugs are fully winged, but species in many different bug families have shortened wings that are called brachypterous or micropterous, or they're even completely apterous. In contrast, only very few true bug taxa have forewings that are hardened and look like a shell that is similar to the forewings, the elytra in beetles, and we call them elytriform wings in the true bugs. In the other 5 true bug clades, where some species have elytriform wings, this wing type is likely originated once or a few times maximum. But this seems to be different in the Dipsacromorpha, where elytriform wings have been described in a number of different genera in the Ceratocombidae and Schizopteridae. So we decided to investigate how many times elytriform wings really originated in Dipsos. We first looked at transitions between macropterous and non-macropterous wing types, such as brachypterous or micropterous, separated by males and females. Both characters were included in the combined data set for Dipsacromorpha I showed earlier. I should also mention that all elytriform types were coded as brachypterous, but a number of the brachypterous wings are not actually elytriform. We found that based on this data set, there were at least 12 changes from macropterous to brachypterous in males, but 21 to brachypterous or micropterous in the females. Looking at the green for males and purple for female symbols on the tree, it is clear that non-macroptery in males and in females is associated in many, but not in all cases. Interestingly, we showed that in several clades, those with orange arrows, the common ancestor had brachypterous females, but macropterous males. And within these clades, males in some, but not all descendant species became subsequently brachypterous. Looking at only those brachypterous wing types that are elytriform in at least one of the sexes, we inferred at least 15 transitions from macropterous or brachypterous to elytriform across the tree. Looking at the elytriform wings more closely, some have traces of wing veins, others do not. Some have smooth surfaces, and others have a lot of small pits on the elytra. So we were curious to see if there were transitions between these types of sculpture and the presence of wings among the elytriform wings. We found that smooth types with wing traces are most common and detected no transition between the different types of elytriform wings. Of course, we would love to better understand what may have driven the repeated evolution of elytriform wings in Dipsos. We are wondering if the occurrence of elytriform wings could be associated with particular microhabitats, even maybe particular microhabitats within, let's say, the leaf litter. Alternatively, or also maybe, the elytriform wings cover the female genitalic structures, so mating can really only happen if the female lifts her wings. We're therefore wondering if in groups where only females are elytriform and where males and females seem to occur in the same microhabitat, elytriform wings may be involved and important during mating behaviors. A quick excursion into an entirely different body part head that in different species of Nanochorus ranges from about as long as high to more than twice as long as high. Even the modestly elongated head shapes in Nanochorus are very unusual for Dipsos, where almost all other species have heads that are distinctly higher than long, as you see up here on the upper right. We generated a preliminary molecular phylogeny of the genus with decent representation of various head shapes, measured head length and height, and reconstructed ratios on the tree, showing that extreme head length, defined by us as more than twice as long as high, evolved independently in the 2 species indicated on the left with arrows. looking more closely at which part of the head is elongated, and something we didn't really actually notice before we started looking into things more closely, is we found that it is really different. What happens is really different in the two species. The gena here on the upper right in green is long in species 1, but it is really the clypeal buccal region that is elongated in species 2. Of course, there are no direct observations of feeding behaviors of these tiny non-occur species at this point, but we speculate that these elongated heads, together with the really unusually long and thin mouth parts in non-occurs, play a role in their predatory behaviors. Finally, structures that are being referred to as male-specific organs, short MSOs, that we have documented in many species across Schizopteridae. They may be even more enigmatic than the wing and head structures I just talked about, but our morphological examinations have shown that all seem to involve either complex glands or glandular trichomes, most likely exocrine glands for uncharacterized compounds. And as the name suggests, they're only found in the males. They are located on different body parts and have different fine structure. So it was pretty clear from the onset that they are not necessarily homologous across the taxa in which they are found. Accordingly, we coded them as 7 separate characters in our combined phylogeny across dipsos based on position and structure, but we also optimized a combined MSO character shown here on the left, mostly to show the overall distributions of MSOs on the tree. They are restricted to the subfamily Schizopteridae. Somewhat unsurprisingly, I want to say most MSO are restricted to an synapomorphies of smaller clades, but the deceptively similar looking wing MSOs in 2 genera were optimized as independently derived, which was a bit of a surprise. Initially, I speculated that there may be an association between presence of MSOs and low species level male genitalic diversity in a given group, which could point towards an involvement of gland compounds in male-female recognition. But this seems to be only the case in 1 or 2 of the clades in which we find MSOs and not the others. And things get really crazy in the Corixidae genus group, for which we have the fairly well sampled molecular phylogeny I showed earlier. MSOs are found in 6 different positions on the body and the forewing, but again, the underlying fine structure of MSOs are similar in consisting of glands or trichomes, and there's at most 1 of the 6 different types of MSOs present in a given species. We did ancestral character state reconstructions of separate binary characters, and all of them combined in a multi-state character. And reconstructions, and the second reconstruction is shown here on the right. And it is pretty clear that things are complicated. While, for example, the frontal MSO that is here shown in blue is restricted to only 1 clade and present in all species, that a different type of MSO that we coded as the anterior pronodal lobe MSO that is shown here in yellow are independently derived in most of the taxa in which they occur. The pattern is particularly intriguing within clade 1 up here on top, where species either have MSOs on the anterior or the posterior lobe of the pronotum or on the forewing, so distinctly different body parts. Assuming that these 3 different MSOs are not homologous, this would mean a lot of gains and losses, even just within these, this relatively small clade. But if one alternatively assumed that they are homologous, one would have to conclude that MSOs switch their position on the body during evolution. And to me, either one of these hypotheses sound pretty crazy, to be frank. So why do I still care about MSOs after all that work we have done on them? Of course, there are still lots of undescribed species, especially given that we have barely touched the tropical and oriental regions, but it is really the crazy morphology of species in this group that keep us going. Just look at the modifications of the mouthparts in these two closely related species. And of course, the hope that one day we'll be able to actually get glimpses into the biology and the behavior of critters in this group. With this, there are, of course, many colleagues to acknowledge, in particular, students and researchers who worked in my lab over the last few years and who became as excited about Dipsos as I am. And I would like to thank you for your attention.
Thank you very much, Christiane. Hi, can you hear? You are mute, I think, Christiane. Hola, hola.
We can hear you.
I can hear you now.
Yeah. You should stop the recording though, please.